Good morning, everyone, and welcome to the PNW Farmers Network Soil Health Coffee Hour. Thanks so much to our speakers for taking the time to come and share some of their work with us on soil arthropods and pollinators in cover crops. It's really exciting to be thinking about the range of different insect life and soil biology beyond the microbes that we typically think about as soil biology. So thanks so much for our guests, Dane Elmquist and Dr. Subodh Adhikari. With that, I'll hand it over to our speakers.
All right. Thanks, Carol. We are excited to be here today. As Carol mentioned, my name is Dane Elmquist. I'm a PhD candidate working with Dr. Sanford Eigenbrod at the University of Idaho, here with Dr. Subodh Adhikari, who's a research assistant professor at the University of Idaho. We are both in the entomology department and are happy to be here to talk to you guys today and give you a bug's eye view of cover crops on the Palouse, on pollinators and soil arthropods. All right, so the inland Pacific Northwest is really a landscape in transition. So wheat fallow is the predominant rotation in the region, but growers, as we know, are starting to convert part of their fallow fields into cover crops or alternative crops. And the agricultural landscape of this region is becoming more and more diverse. And so there are several benefits to agricultural diversification. I think reducing fallow and increasing the sustainability and soil health of those fields is a really big one. And replacing fallow can also intensify production with another cash crop, or for example, introducing a forage crop for livestock grazing. So diverse agroecosystems and cover crops specifically promote soil health. They can aid with weed suppression and pest regulation, as well as promote biodiversity both above and below ground, along with biodiversity's associated ecosystem services. And all of these things ultimately enhance the resilience of our agroecosystems to an increasingly variable environment. So as we mentioned, cover crops are one diversification tool and are of course the subject of today's talks. So cover cropping has several well-recognized agroecological benefits, as you can kind of see on this cover crop wheel on this slide here. But incorporation of cover crops into rotations here on the Palouse is relatively novel, and there's still much to learn, especially about how cover crops impact insects and their ecosystem services. And so that is what Subodh and I are interested in and have spent some time researching over the past several years. And we're starting to learn more and more about this aspect of cover cropping. And today we're going to share some of that research with you. I'm going to get us started here talking about soil arthropods and cover crops, and then Subodh will cover the above ground insect component. All right, so getting into it, soil arthropods. Soil arthropods are a really major component of below ground biodiversity, but I don't think they don't get as much love sometimes as their earthworm and soil microbe counterparts. They really encompass a really wide variety of functional groups like predators, which include things like pseudoscorpions, beetle larvae and adults, as well as predatory mites. There are also herbivores like the familiar wireworm, garden centipedes, as well as decomposers and microbe feeders. This is the kind of predominant functional group of our soil arthropods, and these include things like orobatid or box mites, springtails, and sosids. And yeah, as I mentioned, this group is rather understudied and is really not often considered in management decisions. So it's a fun place to be researching right now. And we're really interested in how they're responding to cover cropping. So our research questions today are primarily focus on the effects of cover crop mixtures. So cover crops are typically grown as single species or two species mixes, but we know that many growers in our region are already adopting or are starting to experiment with more diverse mixes. So in terms of our below ground research questions, to that effect, we set up an experiment to better understand how cover crop mixes affect soil arthropods below ground by asking two different questions, excuse me. Does cover crop diversity above ground affect soil arthropod biodiversity below ground? And we're also interested in understanding, what are the legacy effects of cover crops on soil arthropods and their associated ecosystem services in the following wheat crop? So to answer these questions, we set up a replicated small plot study in collaboration with the Strickland Lab at the University of Idaho. They're interested in biogeochemical cycling and microbial ecology, but we teamed up with them to add an arthropod component to this research. So we examined how 4 monoculture cover crops, pea, clover, sunflower, and flax, as well as a mix of all those monoculture species, affected soil arthropods, compared to each other, as well as a fallow control. And so jumping, right into our results here, um, we measured arthropod communities using this experimental design for 3 years. And we found that, in short, diversity above ground drove diversity below ground. So these graphs here that we're going to look at show the cover crop treatments on our x-axis here, and the richness, which is the number of species, on the y-axis of this left graph, and the diversity, which is the number of species, but also includes evenness in their communities on this graph right here. And so we generally saw that polycultures, shown in purple, on average enhanced soil arthropod biodiversity more than the monoculture cover crops, shown here in orange. And polycultures always outperformed fallow, which is shown here in green. But that wasn't always the case for monocultures. So in short, again, this diversity above ground begets diversity below ground. But again, we're interested in these legacy effects as well, and understanding if these effects on our below ground communities actually persist in the following wheat crop. So to answer this question, we planted all of these small plots to winter wheat in November of 2021. And outside of learning if the effects on biodiversity persisted into the winter wheat crops, we were also interested in learning if the actual functions of the soil arthropod communities were affected by our previous cover crop treatments, because the changes in biodiversity can often lead to changes in ecosystem functioning. So to get at this question, we measured the decomposition rate of a standardized litter substrate using a 240-day litter bag study in the winter wheat plots. So we used 2 litter bag mesh sizes, litter bags that either allowed soil arthropods to access the litter or excluded soil arthropods from the litter. And decomposition is a really important function of our soil arthropod communities, and it's especially relevant in no-till systems where residue is left on the soil surface and it needs to be incorporated and kind of get that nutrient cycling started. And arthropods are a really important component of this process. And I could talk about how important arthropods are to decomposition, but I'll just show you a video that does a better job than I could. So let's just take a second and watch that. Okay, so here we are looking at 15 weeks of decomposition in 20 seconds. So the left side of the screen here shows decomposition in the absence of soil fauna, and the right side is decomposition in the presence of soil fauna. And so already you can see that there's a really big difference in decomposition rate when soil fauna are involved in the decomposition process. So let's take a look at some of our results from our legacy effect experiment. So here we're looking at a graph of the decomposition rate of litter on the y-axis here, and our previous cover crop treatments on the x-axis. Just a reminder, all of this was conducted in winter wheat following our cover crop treatments. So the blue boxes that you can see in this figure are the litter bags that allowed arthropods to access the litter, and the white boxes are litter bags that excluded arthropods. And so, similar to what you just saw in that video, decomposition rate increased when arthropods were allowed to access the litter. Those blue boxes are much higher than those white boxes, but all cover crops were not really equal in their effects on arthropod-mediated decomposition rate, and we observed that a higher arthropod-mediated decomposition rate when a polyculture cover crop preceded winter wheat compared to the monocultures. And so we also, of course, looked at the yield of winter wheat following our different cover crop treatments. And that's what this graph on the left is showing here. So yield is on our y-axis and our previous cover crop treatments, again, on the x-axis. And we saw that the polyculture cover crop increased winter wheat yield significantly more than the flax and the clover monocultures. And this was, numerically, but not statistically, greater than fallow and the other 2 monoculture crops that we used in our experiment. So furthermore, this arthropod mediated decomposition rate, which is shown here in this graph on the right, here on the y-axis, was really positively related to yield across all of our cover crop treatments, with the highest values corresponding to the polyculture cover crop. So just to conclude the below ground portion of this talk, our results really show that cover crops had effects on soil arthropods with greater diversity in our polyculture cover crop treatment. Cover crops also exhibited legacy effects on soil arthropod communities that had functional implications in the subsequent wheat crop, which ultimately contributed to greater litter decomposition rates as well as wheat productivity. So, in short, diversified cover crops can promote soil arthropod diversity as well as their associated ecosystem services that are important for agroecosystem performance and sustainability. So with that, I will turn it over to Subodh and we will go above ground.
Right. Thank you so much, Dan, for leading this and talking about below ground. Let's move to above ground. All right. Thank you, everybody, for coming to this talk again. So now life begins on soil, slowly comes up. So now we started from soil below ground. Now we are coming up. And I'm going to talk about above ground insects. So for this, what we did was we mostly focused on the effect of cover crops on pollinators, insect pests, as well as natural enemies. And for that, first, our research question was, how does this crop mix, diversity of mix, as well as cover crop biomass can impact insect pests, predators, and pollinators at the same time also for weed suppression, because we believe that soil health is the number one for cover cropping in the region, but second to that would be either weed suppression or insect pest management. So for this, we visited 26 cover crop farms in the region, in Washington and Idaho, in our region, in 2019 and 2020. We did sweep net sampling to sample insects, and then also collected some cover crop as well as weed biomass from those fields. And the further results, what we found was the growers on our region, they have been using 35 different cover crop cultivars in their cover crop mixes, ranging from as low as a single crop as a cover crop to up to 20 different crop mixes in the cover crop. And these cover crops are heavily dominated by including brassicas, mostly radish or turnip, purple top turnip. They were very common, but also growers are using legumes, grasses, and several other crops in their mix. And then when we collected weed and weed biomass, we identified about 45 species, and they were mostly dominated by volunteer alfalfa, common lamb's quarter, red root pigweed, mayweed chamomile is another big, and then cheatgrass in the region. Also insects, but this particular diagram looks a little bit complicated, but it's not. I will just walk you through it. So on top of this, you see crop diversity, which is again the crop mix, number of crops used in the cover crop mixes. On the left top is cover crop biomass. Second row, you see weed biomass. And then on the right in the second row is weed richness, so number of weed species there. At the bottom, we see predators, which are natural enemies, like lady beetles, insect pests, as well as pollinators. So what we see here is first, the crop diversity, as you see these colors here, green colors here, and then predators, the natural enemies, the beneficial insects, they were strongly positively associated with the diversity of crops used in the crop mix. So higher diversity of crop mix, better for natural enemies, but which is also better for pollinators, another group of beneficial insects. They were also positively associated with the high diversity of crop mixes there. However, we didn't see any response of insect pests. Insect pests, they were dominated by some small-sized aphids and thrips, but they were not necessarily associated with crop diversity. They didn't really care whether there was like just one or few crops or a high diversity mix. Interestingly, we found that crop biomass, or the cover crop biomass, was negatively associated with the weed biomass. So meaning that if there was a very good standing crop biomass, no matter whether there were too many crops or just a single crop, that was able to suppress weed on their farm. Weed, the crop diversity or the mix was also somehow impacting weed richness, but that was not really significant. So this is our overall result from this above ground. So now, summary of our talk today, what we found. So low biomass of cover crops, high biomass of weeds, high biomass, low biomass of weed. So a good standing cover crop suppresses weed. Low diversity, not very good for pollinators, but high diversity, useful supporting pollinators, but also natural enemies above ground as well as below ground, other beneficial insects, natural enemies, beneficial arthropods below ground. The high mixed diversity was good for both above ground and below ground beneficial insects in that sense. And also, as Dan mentioned, about legacy effect, that after a low diversity cover crop mix, when wheat was planted on that those fields, the decomposition from soil arthropods was slower, lower compared to on a field where the wheat was planted after high diversity cover crops. There was a high decomposition, meaning there were more below ground arthropods there, so beneficial insects there. So the take-home message is our multiple studies together, they show that cover cropping can promote biodiversity-based ecosystem services, both above ground as well as below ground, and specifically suppressing weeds above ground, supporting pollinators and natural enemies above ground, and also increasing litter decomposition, increasing below ground soil arthropods, as well as improving overall soil health. And then, so we thought that this approach of studying both above ground and below ground in a holistic way provides a good picture of the system that we have been working on. So with that, I would like to thank iGovernment Lab, several other people listed here, and then funding agencies that supported our studies. And also would like to thank all the participants today. Any questions? We are happy to answer.